Welcome to ShineTech's YouTube channel, and in this video I'm going to talk about the biosynthesis of thyroid hormones by the thyroid gland. So the contents of this video, we are going to talk about the functional anatomy of the thyroid gland. Then from there we're going to move on to the biosynthesis and storage of thyroid hormones and secretion of thyroid hormones, and finish with transportation of the thyroid hormones. So let's begin by looking at the functional anatomy of the thyroid hormone. So the thyroid gland. So the thyroid gland is the largest endocrine gland, and it weighs about 15 to 20 grams in adults. It consists of two lobes connected by the isthmus, and it is located on either side of the trachea below the larynx. So this is our thyroid gland. It is located in the anterior neck, and it is butterfly shaped. Okay, it has two lobes. We have the left and the right lobe, and we also have it's connected by the isthmus, and it is just on either side of the trachea below the larynx. So to be specific, it is below the cartilages of the larynx, which are the thyroid cartilage, that big cartilage there. Then we also have the cricoid cartilage just below the thyroid cartilage of the larynx. So on the histology part, when you zoom in the lobes of the thyroid gland, you are going to find what we call folliculus, the follicles. So it has follicles. Those follicles are made up of what we call the follicular cells. We have that space there which has homogeneous material, which is called the choroid. So that space there, it has a material, and it is called the colloid. And then we have the basement membrane. Then we have other cells which are near the follicles, which are called the parafollicular cells, or the C cells. Then we also have the capillaries which are surrounding the follicles. Okay, so that's the histology. Now let's look at the biosynthesis of thyroid hormones. So there are two principal thyroid hormones. We have the thyroxine, which is T4, and we also have the triiodothyronine, which is T3. Okay, now these hormones are synthesized from tyrosine and iodine. So tyrosine is an amino acid. Then we also have an ion there, which is iodide. That's where they are formed. But before we look at the biosynthesis, let's understand what stimulates the thyroid gland to start the biosynthesis of thyroid hormones. So the process starts like this. We have the paraventricular nucleus. We know the hypothalamus. That is the part of the hypothalamus, and we have the pituitary gland. So the hypothalamus, it is made up of cell bodies, group of cell bodies, which are called nucleus. So those nuclei, we have a certain nuclei which is called the paraventricular nucleus. So that paraventricular nucleus is going to secrete what we call thyrotropin releasing hormone there. Thyrotropin releasing hormone is going to travel. Okay, so that is the nucleus there. And then we have axons. So the axons are going to travel from that far end into the pituitary gland, specifically the anterior pituitary gland, okay, where it is going to form a dilated part, which is the terminal part of the axon. Okay, so that part, that's where the secretion of what we call thyrotropin releasing hormone is going to be stored. Once it's stimulated, it's going to release thyrotropin releasing hormone into the pituitary gland, stimulating the pituitary gland to release other hormones, which we call thyroid stimulating hormone. That thyroid stimulating hormone released by the pituitary gland is going to stimulate now our thyroid gland to release or to start the biosynthesis of our thyroid hormones. Okay, so that thyroid is thyrotropin releasing hormone can travel in two parts. Okay, there are two ways in which hormones, which from the hypothalamus goes into the pituitary gland. So we have the pituitary here, and then we have our hypothalamus above there. So these are the neurons from the hypothalamus, okay, into our pituitary gland. Okay, so the hormones can travel by two ways. Remember, they are synthesized by the nucleus there. Then they travel to the terminal part of the axon, where they are released. So we have the first tract, which is called the tubulo-infundibular tract, and the hypothalamic hypophyseal portal system. This system here, it is the tubulo-infundibular tract and the hypothalamic hypophyseal portal system. Then we have another system which is called the hypothalamic hypophyseal tract. Okay, so this one is simply direct from the hypothalamus direct into the pituitary gland. Okay, and then in the release of hormones is just direct into the capillary there. But here we have the connection of the neuron and the portal system. Now the portal system is going to facilitate the movement of those hormones into the anterior pituitary gland. So our thyrotropin releasing hormone is going to be secreted by this neuron here. Then once secreted there, it is going to go into the bloodstream. Then the blood in the portal system, which is the long portal vein into the anterior pituitary gland. The anterior pituitary gland is going to be stimulated to release thyroid stimulating hormone, which are going to go to the thyroid gland to initiate the biosynthesis of thyroid hormones. Okay, so that's what happens. So the tract which is used is the tubulo-infundibular tract and the hypothalamic hypophyseal portal system. Now let's now dive into the biosynthesis of thyroid hormones. Remember here now it has been stimulated by the thyroid stimulating hormone from the pituitary gland. So let's divide it into 5 steps. The first step, we need our iodine. Remember we're saying we need tyrosine and iodine molecule. These, they are going to form our thyroid hormones. So let's divide it into 5 steps. The first step is iodine trapping. The second step is the synthesis and secretion of our thyroglobulin. Remember, thyroglobulin is simply a big molecule of tyrosine. Okay, and then the third step is oxidation of iodide, and then fourth step is organization of thyroglobulin, and finally coupling reaction, which will lead to the formation of thyroid hormones. Okay, so iodine trapping. So under iodine trapping, remember we in iodinated salt or in other sources, we have what we call iodide ions. So those iodide ions are absorbed in the bloodstream. So they're going to travel in the bloodstream. Now, when they reach the thyroid gland is going to trap those iodide ions via a iodide pump. So this here is what we call the iodide pump. The other name is sodium iodide symporter. So this sodium iodide symporter is going to trap our iodide ions. Okay, so remember it is a symporter. It is going to transport 2 sodium for 1 iodide. Okay, iodide ion. So that's how they are trapped. Then from here, our iodide ions are going to be transported into the choroid. Now in the choroid, that's where all the reactions which leads to thyroid hormone formation takes place. So the transportation of iodide from the follicular cells into the choroid is facilitated by the pendrin. So the pendrin is going to help to transport this iodide ion from the follicular cells into the choroid. Okay, so that's what happens under iodine trapping. Okay, so now let's move on to the second step. We have our iodide ion in the lumen of the follicular cells, which is in the choroid. Now we need our protein, which is the thyroglobulin. Remember, thyroglobulin is simply a molecule which is made up of about 140 tyrosine amino acid. Okay, tyrosine. So now let's form our protein. Okay, so our protein is going to be formed when the G-coupled receptor is stimulated by the thyroid stimulating hormone. So the thyroid stimulating hormone is going to bind to our receptor, G-coupled receptor, and now the alpha subunit of the G-coupled receptor, remember it is made up of the gamma, beta, and alpha subunit. Okay, so the alpha subunit is going to slide. Okay, in the first place, it binds GDP, but when it's stimulated, it is going to release that GDP and bind GTP. Okay, then from there it is going to slide over to our adenylyl cyclase. So once it binds to adenylyl cyclase, it is going to activate that adenylyl cyclase, and then adenylyl cyclase is going to convert ATP into cyclic AMP. Now cyclic AMP is going to activate a protein kinase. Now the protein kinase is going to phosphorylate a transcription factor. Transcription factor is going to initiate the transcription of mRNA. Okay, so once mRNA has been transcribed, it's going to be transported to the rough endoplasmic reticulum, where translation is going to take place. Translation of tyrosine is going to take place. Once tyrosine has been translated, has been formed, then it is going to be taken to the Golgi apparatus for prepackaging, and okay, to be sorted and prepackaged. And then from there, we are going to have our thyroglobulin, which is 140 molecules of tyrosine. So it's a big molecule. Okay, so let's see what happens here. Okay, so this is what happens. We have a molecule has binded there. Then this is an alpha subunit of the G-coupled receptor binding to the cyclic AMP, formation of CAMP, protein kinase, transcription there, then transported to the rough endoplasmic, formation of our protein. So that's how thyroglobulin is formed. Okay, so that's how it is formed. So now let's move on to the third step. Third step, now we have oxidation of iodide. So oxidation of iodide. So once our iodide ion has been trapped into the follicular cell, we said it is transported in the choroid by what we call pendulum. So this is a pendulum transporter. So it goes in the lumen, which is the choroid. Now once in the choroid, we have an enzyme which is called peroxidase. Okay, thyroid peroxidase is going to help. Okay, so the peroxidase. So peroxidase is going to facilitate oxidation of iodide. Okay, have that iodide here. So it is going to be oxidized to iodine molecule. Okay, by the removal of an electron to a molecule. Okay, with the oxidation number 0. Okay, so that's what happens under oxidation. Remember, the enzyme involved is the peroxidase. Okay, and then from there, we said our protein has been formed by the process which we have just described in the previous step. So that protein now is going to be released in the choroid by exocytosis. So that is a long chain of thyroglobulin which is being released there. Okay, now we have our two substrates. Now we need to form the reaction which will lead to formation of our thyroid hormones. So we have that molecule there, or that complex molecule, which is thyroglobulin. It is going to be released in the choroid by exocytosis. Then we also have our oxidized iodide ion there. Now it is now a molecule. So let's see now what happens under organization of thyroid globulin. So under organization here, what happens is that as I've already said, our protein is going to be released by exocytosis by the follicular cells into the choroid. Now once in the choroid, what happens is that tyrosine, tyrosine is part of it's the one which makes the thyroglobulin, is first now iodinated. Okay, to be iodinated at position 3 to form what we call monoiodotyrosine, which is MIT. So we have one molecule of iodine which is iodinated to the position 3 of our tyrosine to form monoiodotyrosine. Then from there, in the second step, we are going to have another iodine molecule being iodinated to tyrosine at position number 5 to form what we call a di. Remember, di is 2 and mono is simply 1. So here we have diiodotyrosine, which is 2 molecules of iodine. So that's what happens under organization of thyroglobulin. Now, under coupling reaction. So remember, we had a long chain of thyroglobulin. Now, under coupling reaction, our long chain of thyroglobulin has been iodinated. Okay, has been iodinated here. So let's say this is the long chain of thyroglobulin. It has been iodinated with the iodine there. So there we have one, which is monoiodotyrosine. Then here we have two molecules of iodine. So this one is di, because there are two. So this one is diiodotyrosine. Then this is also diiodotyrosine here and also there. So under coupling reaction, what will happen is that our thyroid peroxidase is going to couple in a coupling reaction. Is going to fuse this part with that part. Okay, so we're going to have a monoiodotyrosine with a diiodotyrosine being fused together, okay, in a coupling reaction. So once they fuse together here, let's see what happens. Okay, so they fuse together here. They are going to be coupled together to form a molecule with 3 iodine. Okay, so it is going to form a compound with 3 iodine molecules. So that is what we call triiodothyronine. Okay, the first hormone which we mentioned we are going to form 2 thyroid hormones, which is the thyroxine and T4 and T3. So this one is T3. Under coupling reaction, we had a monoiodotyrosine and a diiodotyrosine coupled together to form T3. Okay, so the second step again is to form our T4. So our T4 is formed when a di, which has 2 iodine molecule and another di coupled together to form T4. So remember, this is T3. Okay, so this is T3, and now we are forming our T4. So our T4 is going to be formed when a diiodotyrosine is coupled with a diiodotyrosine. So remember, they are still connected by this chain here. So it's a long chain. We are going to have our T3 here, and we are going to have our T4 there. So this is how these hormones are formed under coupling reaction. Okay, so now storage. We have formed our hormones, which is T3 and T4. Remember, they are still connected. So storage is simply they are stored in the lumen of the follicle as choroid. So that choroid we are talking about is simply the same hormones which have been stored there for several months. Okay, they are stored there for several months, but when stimulated, they can be released. That's how they are stored. Okay, so remember, we said we are going to look at the content. On the content, we said we are going to look at the functional anatomy, now which we are done, and then we have looked also at the biosynthesis. Now let's look at the secretion and transport. So under secretion is divided into two. We have endocytosis and proteolysis. Remember, we said that hormones are stored inside the follicle, inside here. That's where they are stored. So to remove them from that, from inside the follicle, the choroid there, we need endocytosis. So what happens? The lumen, the apical surface of the follicular cell, they are going to endocytose those hormones, okay, to form what we call the choroid droplets. Okay, so choroid droplets are going to be formed here. And remember, this process is facilitated by what we call a TG receptor megalin. Okay, megalin. So this receptor is just on the apical surface there. That's where it is. So it's going to facilitate the endocytosis of our hormones to form what we call the choroid droplets there. So once our choroid droplets have been formed, endocytosis is done. Now we move on to proteolysis. So proteolysis, what happens is that we have our lysosomes inside the follicular cell. Remember, this is a follicular cell. So lysosomes are going to come, and then they are going to fuse with that vesicle there to form one vesicle where we have our hormones and the enzyme proteases. Proteases are the enzymes in the liposomes which fuse with that choroid droplet containing the hormones. So once proteases fuse with our choroid droplets, what will happen is that long chain we formed here, they are going to be cleaved here, they are going to be cut. Okay, so we are going to cut this. So the proteases are going to cut, the enzymes in the liposomes are going to cut to form two hormones, which is T3 here and T4. Okay, so that's what happens under secretion. So once it has been secreted here, once it has been cleaved, once the proteases have worked on the long chain of thyroglobulin to form T3 and T4, now what will happen is that those hormones are going to diffuse in the bloodstream on the lumen there. They're just going to diffuse. So here on the apical surface we have endocytosis, but on the basal surface there we have diffusion. That's how they are secreted. Okay, but before they diffuse, proteolysis has to take place. The lysosomes have to fuse, and then proteases has to cut the long chain of thyroglobulin, iodinated thyroglobulin, into T3 and T4. Okay, that's under secretion. So let's move on to transportation, this part here, the last part. So under transportation, now we have our T3 and T4. What happens is that under transportation, these hormones are transported as bound to certain proteins or in free form or unbound. So under bound, we have these hormones circulate bound to specific binding proteins. So the first binding protein, which transports about 70%, is thyroxine binding globulin. Thyroxine binding globulin is going to bind about 70% of those hormones. Then another one have thyroxine binding prealbumin, which is going to transport 15 to 20%, and then we have thyroxine binding albumin, which is going to transport 10% of T4, and in free form we only have a little amount, which is 0.05% of T4 is going to be transported as free form, not bound to anything, and then we also have 0.5% of T3 is going to be transported and bound. So these are considered active. These are the ones which are active. The ones which are transported as bound, they are not active up until they are unbound. Okay, so thank you so much for watching this video. If you like this video, please hit that like button there, and also don't forget to like and hit that notification bell, and leave your comment please concerning this video.